The field of anti-aging research has made extraordinary strides in recent years, with dramatic results emerging from laboratory experiments, particularly in short-lived animals like mice, worms, and flies. Interventions such as caloric restriction, senolytics, N80+ precursors, rapamycin, and gene editing have repeatedly extended lifespan and improve health span in these models, sometimes by as much as 30% to 50%. These results are not trivial. They point to biological pathways that are deeply conserved across species and suggest that aging is not an immutable fate, but a malleable process. Yet, as promising as these breakthroughs are in controlled lab environments, the challenge of translating them into effective human therapies remains steep and often frustrating. One of the primary barriers lies in the simple biological fact that humans are not mice. While many aging mechanisms, such as oxidative stress, mitochondrial dysfunction, or telomere shortening, appear across species, the complexity, longevity, and variability of the human body make direct extrapolation risky. A mouse lives for about 2 years. Even if a drug doubles its lifespan, the long-term effects can be observed quickly. In humans, observing such effects might take decades. That means researchers must rely on indirect measures of aging, such as epigenetic clocks, inflammatory biomarkers, or muscle strength, all of which may be useful but are still proxies for actual lifespan and late-life disease burden. Furthermore, the genetic diversity of humans adds another layer of complexity. Lab animals are typically genetically identical and housed in strictly controlled environments. Humans, by contrast, live highly variable lives, exposed to different diets, pathogens, pollutants, stressors, and social environments. An anti-aging compound that works under ideal lab conditions may behave unpredictably in the real world, especially when interacting with the unpredictable variables of lifestyle, environment, and coexisting health conditions. A molecule that slows aging in a lean, young lab mouse may have very different effects in an overweight, sedentary 70-year-old human with hypertension and prediabetes. Clinical trials are the natural next step to validate findings, but they face enormous structural and ethical hurdles. First, there is the issue of duration. Most human clinical trials last months or a few years, far too short to capture the full impact of an anti-aging therapy meant to work over decades. This leads researchers to focus on short-term surrogate markers like insulin sensitivity or inflammatory profiles, rather than actual incidence of Alzheimer's, heart disease, or mortality. While useful, these proxies can sometimes mislead. A drug that improves a marker today may have unknown long-term side effects that only become apparent years down the line. Another major limitation is funding. Unlike cancer or diabetes research, which attracts billions in public and private investment, anti-aging science still sits in a gray area. Aging is not officially classified as a disease, which makes it harder to receive grants or insurance reimbursement. Pharmaceutical companies often hesitate to invest in aging research unless it targets a specific condition like osteoporosis or cognitive decline. The result is a fragmented field that struggles to scale promising lab findings into robust, human-centric trials that can meet regulatory standards. Participant diversity also presents a critical barrier. Many trials in aging research are conducted on relatively healthy older adults, often white, middle-class individuals living in urban centers. But aging plays out differently across socioeconomic, ethnic, and gender lines. Disparities in access to care, nutrition, and stress all shape how the aging process unfolds, and these variables must be accounted for if we hope to develop therapies that work for the general population. Without broader participation, we risk developing interventions that benefit only a narrow slice of humanity while leaving others behind. Regulatory ambiguity adds yet another layer of difficulty. The FDA does not currently recognize aging itself as a treatable condition, meaning any anti-aging therapy must be tested as a treatment for a specific disease or symptom, such as arthritis, insulin resistance, or frailty. This regulatory model complicates both the design and approval of clinical trials. It also creates disincentives for companies to market truly preventive therapies, pushing them instead toward managing late-stage symptoms of aging-related diseases rather than targeting root causes. Despite these challenges, progress is being made. New frameworks for measuring biological age, such as DNA methylation clocks and comprehensive biomarker panels, are helping to quantify aging in ways that can be used in trials. Initiatives like the TAME, Targeting Aging with Metformin study aim to create a blueprint for testing anti-aging drugs in a clinical context. Longitudinal cohort studies are beginning to identify which interventions, whether diet, exercise, supplements, or pharmacological agents, correlate with slower biological aging. And the biotech world is increasingly engaging with longevity science, bringing new energy, capital, and urgency to the field. Ultimately, translating anti-aging research from lab to life is not just a technical challenge, it's a cultural and institutional one. It requires us to shift how we think about aging, not as an inevitable decline, but as a process that can be understood, measured, and potentially modified. It also demands a new kind of patience, ironically, in a field devoted to defying time. The breakthroughs in mice are real and promising, but the road to meaningful human application will require sustained investment, interdisciplinary collaboration, ethical oversight, and above all, the willingness to dream beyond the lab bench. If we can overcome these barriers, the future of anti-aging medicine could be transformative, not just in extending life, but in extending the years of life that are healthy, independent, and fulfilling. That vision, though still unfolding, is worth every ounce of scientific effort and societal will.